Hello, my name is Mike Galluzzo. I'm a PhD student at UC Berkeley, and thank you for coming to my virtual talk, which I've titled Probing the Dynamic Structure of Block Copolymer Electrolytes Under DC Polarization with In Situ Small Angle X-ray Scattering. The motivation for my research is to address challenges for next generation lithium metal batteries. The current state-of-the-art lithium-ion battery typically consists of a transition metal oxide cathode separated from a graphite anode by this pore separator, which is wetted with a liquid electrolyte LiPF6 and ECDMC. And now the graphite anode intercalates lithium ions where 6 carbon atoms are necessary per lithium ion to house the lithium. If we can instead use lithium metal directly on a copper current collector, we could increase the energy density of this battery. However, with traditional liquid electrolytes, this results in the formation of dendrites, which can short circuit the cell and ignite the flammable liquid electrolyte. And so, while lithium metal offers increased energy density, it is not compatible with typical lithium-ion electrolytes. One potential solution to this problem is to use composite electrolytes to enable the lithium metal anode. In this schematic, I'm showing a polystyrene block polyethylene oxide block copolymer electrolyte, or SEO for short, where the two polymer blocks microphase separate into polystyrene-rich domains, which provide mechanical rigidity but do not conduct ions, and polyethylene oxide-rich domains, which solvate a lithium salt, in this case LiTFSI. And for the purposes of this talk, we'll define this molar salt concentration R, which is the molar ratio of lithium ions to ethylene oxide moieties. And in this polyethylene oxide salt-rich domains, ions are transported between the anode and cathode of the cell. Now, it's been shown in the literature and through a number of patents that these block copolymer electrolytes are capable of enabling a lithium metal battery, or at least operating in a battery containing a lithium metal anode. However, it is not clear in the literature how a block copolymer electrolyte facilitates ion transport at high current densities. And so this is the question I'd like to focus on for the rest of the talk. The reason this question is important is because at high current densities, significant salt concentration gradients will develop across the electrolyte. Here I'm plotting the local salt concentration as a function of position in the cell, and if we apply potential across the electrolyte, anions will be attracted to the right and cations to the left in the direction of the electric field, but because anions cannot react at the electrode and electrode neutrality must be maintained, this results in the formation of a salt concentration gradient across the electrolyte. And if we increase the potential or increase the current density, this results in an increase in the salt concentration gradient, where we expect a maximum value to be about twice the average salt concentration because the total salt concentration in the electrolyte must be conserved. Now, the formation of these salt concentration gradients is not too much of an issue for traditional liquid electrolytes because the solvent can easily flow and rearrange to accommodate the salt concentration gradient. However, in a block copolymer electrolyte where salt only resides in the PEO phase, it is not clear how the presence of rigid polystyrene domains might affect this rearrangement. In particular, the rigidity of these domains are meant to suppress the flow of the polymer to prevent dendrites. And so the question we want to answer is how the formation of salt concentration gradients during polarization influence the morphology and flow of the polymer. So to begin to answer these questions, we first want to consider the magnitude of the salt concentration gradients which forms. And to do this, we can apply Newman's concentrated solution theory, which starts with the Stefan-Maxwell diffusion equations applied to a concentrated liquid electrolyte. From here, we can recast these equations into measurable quantities and then further derive these integral relationships, which can yield the cell concentration profile as a function of position in the cell when these four transport parameters are known as a function of salt concentration. And while I don't want to belabor these points or these measurements, I just want to say that we've measured these parameters. Here I'm showing the connectivity as a function of salt concentration for a variety of different block copolymer SEO electrolytes with different morphologies and chain lengths. And we've also measured the diffusion coefficient and the current fraction rho plus and the transference number and thermodynamic factor for all of these systems so that we can use this theory in order to predict the salt concentration profiles based on Newman's concentrated solution theory. And all of these measurements are presented in this paper, but I don't want to spend any more time talking about that and instead move on to discuss how we can test these model predictions. So in order to test these model predictions, I've developed a cell in order to use small angle X-ray scattering to study the morphology of the block copolymer electrolyte during DC polarization. The cell is shown schematically on the left and a picture of the cell is on the right. We simplify the cell by using 2 lithium electrodes, which we can apply a potential across to pass DC current, and then we raster the X-ray beam across 3 different positions in order to measure the small angle X-ray scattering patterns shown here to study the morphology of the electrolyte as a function of position during polarization. So in order to make sense of the results of this experiment, we first want to understand the morphology of the block copolymer electrolyte as a function of salt concentration in the quiescent state. And so here I'm plotting the phase behavior as a function of the average salt concentration in the electrolyte. At low salt concentrations and high temperatures, we observe a disordered morphology where the polystyrene and the polyethylene blocks are homogeneously mixed. But if we increase the salt concentration or decrease the temperature, we get this lamellar morphology in the blue region. And if we further increase the salt concentration, we get the gyroid phase in the green region. And on the right axis, I'm plotting the domain spacing, which is also measured from small angle X-ray scattering as a function of salt concentration. And we see that in this region, it follows a predictable trend, which we can fit to this double exponential function in order to obtain a relationship for the domain spacing as a function of salt concentration. And this implies that we might be able to use the domain spacing and morphology to serve as a proxy for the local salt concentration in the cell. And in particular, we pick one salt concentration to use for this experiment, which is shown as a filled circle and is near the disorder to order phase boundary. And so an open question is whether or not this current-induced salt concentration gradient will result in morphological changes in the block copolymer. And just to briefly go over the analysis of the small X-ray scattering patterns, we pass X-rays through our sample and convert the 2D scattering patterns into 1D scattering patterns, where for a disordered morphology, we get a characteristic broad scattering peak, and for a lamellar or ordered morphology, we get the sharp scattering peak, where in both cases, the position of the scattering peak is related to the domain spacing. We're now ready to talk about the results of this electrochemical experiment. Here we applied 500 millivolts per millimeter for 8.3 hours, at which time the salt concentration gradients will build up, shown in red. And after that time, we set the current to 0, switching the cell to open circuit, and those curves will be shown in blue as the salt concentration gradient relaxes. So we can look at the 1D profiles from 3 regions of the cell. Starting at the bottom, at early times, the morphology is initially lamellar, indicated by the sharp scattering peak in the black curve. But as the polarization continues, we see this sharp scattering peak give way to a broad scattering peak indicative of the disordered morphology. And when the cells switch to open circuit and the salt concentration gradient relaxes, we again recover the sharp ordered peak of the lamellar phase. This indicates to us that we do observe morphological changes due to the polarization, and they are reversible. In the middle of the cell, we see a similar behavior. At steady state, we have coexistence of the lamellar and disordered phases, but we again return to the original morphology in the long time limit. The behavior at the salt-rich electrode is more interesting. We expect polarization to result in increase in salt concentration, and And we see the signatures of the lamellar phase throughout the experiment. However, we also observe the formation of these scattering peaks on either side of the primary lamellar peak. And if we go and zoom in on these and index the peaks, they're unambiguously assigned to the gyroid morphology. And if we look at the 2D scattering image, we see that there are these sharp spots of intensity on either side of the lamellar ring, which indicates to us that this gyroid phase forms in relatively few grains. So the next question we want to ask is how these observations compare to the model predictions. And so, as we mentioned, we can use Newman's concentrated solution theory and these equations along with our measurements of the different transport parameters as a function of salt concentration to obtain predictions of the local salt concentration as a function of position in the cell, and then obtain average salt concentrations for each region at steady state at the end of polarization. We can then go back and look at our SACS data and pull out domain spacings as a function of time for the various positions, and then I won't go into the details, but we can use our fits to convert these domain spacing measurements to salt concentration. And that is what I'm showing here, is the experimentally measured local salt concentration as a function of time. And we see that we have fairly good agreement where the solid lines represent the experimental data and the dashed lines represent the theory predicted at steady state. And we obtain fairly reasonable agreement to at least confirm the predicted the magnitude of the predicted salt concentration gradient. So we're now able to compare our experimental results to the theory. So initially, our cell is of uniform salt concentration and in the lamellar phase, shown here. And the salt concentration is near the lamellar to disordered boundary, but is relatively far from the lamellar to gyroid phase boundary shown here. At steady state, after 8.3 hours of polarization, we have some pockets of gyroid phase forming. But if we look at the salt concentration gradient at steady state, which we just confirmed experimentally, we see that the maximum salt concentration is relatively far from what we'd expect to be required to form the gyroid phase. And so it's clear that our one-dimensional models of ion transport cannot capture emergence of the gyroid phase, and there must be another explanation. So we'd like to propose a mechanism for how we observe these pockets of gyroid phases based on the three-dimensional structure of the block copolymer electrolyte. If we consider a defect where the insulating polystyrene domains block the flow of ions in the direction perpendicular to the electrodes, we can imagine that application of an electric field will result in accumulation of salt in this defect site, and if this local salt concentration at this defect exceeds the critical value needed to maintain the gyroid, then we can observe a phase transition to the gyroid morphology. And because these defects aligned in this way are likely to be relatively rare, we only see few pockets emerge of this gyroid morphology. And so our in situ SACS experiments has revealed signatures of concentration hotspots, or local regions of increased salt concentration, which can form in a composite battery material. So I'd like to conclude my talk with a brief summary. We've shown that the in situ small angle X-ray scattering experiments reveal complicated phase behavior in a block copolymer electrolyte when we apply an electric field. We unexpectedly observe pockets of gyroid phase, and we attribute this to the formation of concentration hotspots due to the 3D structure of the block copolymer electrolyte. And what this tells us is that 1D models of ion transport are not sufficient for block copolymer electrolytes, and this work motivates the development of ion transport models which can account for the insulating phase in 2 or 3 dimensions. And I would just finally like to acknowledge my group, the Balsara Lab at UC Berkeley, as well as my advisor, Professor Natasha Balsara, the ALS Beamline 733 staff, where the X-ray scattering experiments were performed, and finally the U.S. Department of Energy Battery Materials Research Program for With that, I'll conclude this talk and just bring up my title slide and say I'm looking forward to answering any questions throughout this virtual conference or over email, galluzzo.m@berkeley.edu. Thank you very much.